Concept 15.4. Alterations of chromosome number or structure cause some genetic disorders. Large-scale chromosomal changes can affect an organism's phenotype. Most of these changes in mammals result in abortion or individuals with developmental disorders. Plants can take these changes better than us. One example of an abnormal chromosome number is non-disjunction. Non-disjunction is when members of a pair of homologous chromosomes do not move apart in meiosis I, or whenever a pair of sister chromatids don't separate during meiosis II.
Now, if either of the aberrant or deviating gametes combines with a normal gamete at fertilization, the zygote will then also have an abnormal number of a particular chromosome. This condition is known as aneuploidy. Now, don't confuse aneuploidy with polyploidy. That is, when some organisms have more than 2 complete chromosome sets in all somatic cells. More specifically in polyploidy is triploidy and tetraploidy. Triploidy indicates 3 chromosomal sets, or 3N, and tetraploidy indicates 4 chromosomal sets, or 4N. How does one attain a triploid cell, you might ask? Easy. The fertilization of an abnormal diploid egg. And tetraploidy can occur from the failure of a 2N zygote to divide after replicating its chromosomes. Following those meiotic divisions, you would end up with a 4N embryo. Now, where in the world can we see polyploidy? Easy, we see it every day. Bananas, wheat, and strawberries. Bananas are triploid, or 3N. Wheat is hexaploid, or 6N. And strawberries are octoploid, or 8N. On to monosomic versus trisomic. Monosomic is the fertilization involving a gamete that has no copy of a particular chromosome will lead to a missing chromosome in the zygote, or 2N-1. That gives you 1 less chromosome than what you should have. Now, trisomic is the exact opposite. Trisomic, if a chromosome in the zygote is triplicate, the cell has 2N plus 1 chromosomes. Through mitosis, the anomaly would be transmitted to all embryonic cells. If the organism even survives, it would have a special set of traits caused by the extra or missing chromosomes. This is how we see trisomy in humans. Down syndrome is one example, and it is called trisomy 21.
Alterations of chromosome structure. Breakage of a chromosome can be caused by errors in meiosis or damaging agents such as radiation. There are 4 types of changes in chromosome structure. The first type, deletion, occurs when a fragment of a chromosome is lost. This means that some of the genes on the chromosome are missing. The second type is duplication. Duplication occurs when a deleted fragment from another chromosome attaches as an extra fragment to a sister chromatid. This causes a certain fragment to be repeated or duplicated on the sister chromatid. The third type of change is an inversion. An inversion is when a chromosomal fragment breaks off and reattaches to the same chromosome, but in the reverse orientation. The fourth and final type of change is a translocation. This occurs when a chromosomal fragment joins a non-homologous chromosome. The most common type of translocation is a reciprocal translocation, in which non-homologous chromosomes exchange fragments. Deletions and duplications usually occur during meiosis. When chromosomes are crossing over, non-sister chromatids will sometimes exchange unequal-sized segments of DNA. This results in one chromosome with a deletion and one with a duplication. A diploid embryo with a large deletion is usually missing many essential genes. This condition could be lethal. Inversions and translocations can alter the phenotype because the gene's expression is influenced by its location with neighboring genes.
Any alteration to the number of chromosomes results in a disorder. The normal number of chromosomes in humans is 46. Non-disjunction and meiosis results in aneuploidy in gametes and any resulting zygotes. Frequency of aneuploid zygotes is high in humans. Most of these chromosomal alterations are so disastrous to the development that affected embryos are spontaneously aborted long before birth. Some types of aneuploidy, however, appear to upset genetic balance less than others that allows individuals with aneuploid conditions to survive birth and live. These individuals have a set of traits called a syndrome. One aneuploid condition is trisomy 21, or more commonly known as Down syndrome. Down syndrome affects 1 in every 700 child born in the United States. The reasoning behind the condition is an extra chromosome, 21, so each cell has 47 total chromosomes instead of 46. As you can see, the one parent experiences non-disjunction, and both their chromosomes go to one cell instead of one going to each cell, which allows 3 chromosomes to be in the 21st chromosome instead of 2. Since the cells are trisomatic for chromosome 21, it is called trisomy 21. People with Down syndrome have characteristics such as facial features, short stature, correctable heart defects, and developmental delays. They are also at a higher risk for leukemia and Alzheimer's disease. However, they have a lower rate of high blood pressure, atherosclerosis, stroke, and many types of solid tumors. With proper treatment, they can live to be middle-aged and live a normal life. Frequency of Down syndrome increases with the age of the mother. At age 30 or less than that, the risk is only 0.04% of having a child with Down syndrome. At age 40 or higher, the risk jumps to 0.92%. This correlation between the age of the mother and the resulting with a child with Down syndrome has not been explained. Most cases results from non-disjunction during meiosis 1. Some research points to an age-dependent abnormality in meiosis checkpoint that normally delays anaphase until all connected cores are attached to the spindle fiber.
In addition to Down syndrome, non-disjunction of sex chromosomes produces many other aneuploid conditions. The reason behind this is the Y chromosome doesn't carry many genes, and extra copies of X chromosomes become bar bodies or inactive on the somatic cells. Aneuploid conditions affect males and females. I'd first like to talk about the syndromes that affect the males. The first syndrome is Klinefelter syndrome. This affects 1 in every 500 to 1,000 male births. This results in an extra X chromosome in the male, XXY. With this condition, males have male sex organs, however testes are abnormally small and the male is sterile. Female characteristics are seen in many cases, even though the second X is inactive. Contrast to XXY, another condition, XYY, affects 1 of 1,000 males. Their development appears normal. In all cases, males are just very tall. Aneuploid conditions affecting females are trisomy X or XXX, which we see in about 1,000 female births. Like XYY, females develop normally other than being taller than average. Triple X women are fertile, but do have the risk of developing some type of learning disability. A more severe condition, monosomy X, or Turner syndrome, affects 1 in 2,500 females. Although phenotypically female, they are sterile because sex organs will not mature. If a female with this condition were to have an estrogen replacement, secondary sex characteristics will develop over time. One last topic to discuss are disorders caused by structurally altered chromosomes. Deletion in human chromosomes causes severe issues. An example of this, Cri-du-chat, or cry of the cat, is when chromosome 5 is erased. Children are born intellectually and physically disabled. Like the name says, the baby's cry is described as a distressed cat meowing. Most infants do not survive with this disease. Chromosomal translocations have been implicated in certain cancers, such as chronic myelogenous leukemia. This occurs when a reciprocal translocation happens during mitosis. Cells will become white blood cells. The large portion of chromosome 22 with a small portion of chromosome 9 produces what is called Philadelphia chromosome. This causes cancer by activating a gene that causes an uncontrollable cell cycle progression.